Hi, everyone. Welcome to Osteobites, and thank you so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and also Director of Scientific Programs at MIB Agents. And today on Osteobites, we are joined by Dr. Yanding Zhao from Stanford University, and he will discuss distinct patterns of chromosomal instability that fuel osteosarcoma progression and influence patient outcomes. Thanks so much, Dr. Zhao, for joining us on Osteobites today. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom, and we will share those with Dr. Zhao after his presentation. A little bit more about our guest today. Dr. Yanding Zhao is a postdoctoral researcher at Stanford University in the lab of Dr. Christina Curtis. He earned his PhD in genetics from Dartmouth College, where he began developing computational tools to understand how genome instability disrupts gene regulation in cancer. At Stanford, his research focuses on pediatric osteosarcoma, and by combining genome sequencing, 3D chromatin mapping, and spatial transcriptomics, he studies how tumors evolve, resist treatment, and evade the immune system. Dr. Zhao works closely with clinicians and scientists to help turn these discoveries into potential new therapies. He's honored to be part of the MIB Agents community and looks forward to sharing his work in a way that resonates with patients, families, and advocates. We're thrilled to have you on Osteobites. Dr. Zhao, thank you so much. Just one announcement before we get started. We are in the thick of September, which is Childhood Cancer Awareness Month. And so you can go gold with us this September by supporting our Gold Bow campaign. Each gold bow is a powerful symbol of hope, resilience, and solidarity for children and families facing cancer. You can help us turn neighborhoods gold and make a meaningful impact, one bow at a time. You can either make a donation to receive a bow or a pack of bows, or you can sign up as a bow agent volunteer. And as a bow agent, you'll help raise awareness by distributing beautiful gold bows throughout your community on mailboxes, storefronts, schools, and more. And I will put a link in the chat where you can get more information about that. And then we'll go ahead and get started with our presentation today. So Dr. Zhao, if you want to go up and pull up your slides.
Sure. And thank you, Christina, for the nice introduction. So can share my work.
That looks great.
Okay. Okay. So hi, everyone, and good afternoon. So today I will show you how different types of chromosomal instability fuel osteosarcoma progression and shift patient outcome. This talk is based on our recent integrative multi-omic study across more than 70 patients. So let me change my point. Okay. So we all know that osteosarcoma is the most common pediatric bone cancer. You can see it peaks in the teenager years right around puberty. So it not only has the highest prevalence, but also hits patients at a very vulnerable stage of life. The standard MAP chemotherapy was established back in the 1980s. It was a big success at the time and clearly improved survival. But since then, there has been almost no progress. Even the largest global phase 3 trial, EUROMAS-1, showed no benefit from adding more drugs. So after 40 years, MAP remains the only frontline option. More recently, immunotherapy was tested. Unfortunately, response rates have been disappointing, as shown here for anti-PD-1 therapy. But if you stratify patients by PD-1 expression, you see a clear difference. PD-1 high patients tend to respond better. This tells us something important. It is not simply that immunotherapy doesn't work. It means better classification matters. Without stratification, we can't match the right therapy to the right patient. So where can we look for better ways to stratify? One unique feature of osteosarcoma is its genome. On the left is a normal genome, tidy and diploid. On the right is the osteosarcoma genome. Chromosomes shattered, rearranged. Oncogenes like MYC scattered across multiple locations. So this is one of the most extreme cases of chromosomal instability in human cancers. And that's why genomic instability may provide a useful way to stratify patients. But genomic instability itself is not a single entity. It is the field for tumor evolution. But different tumors may channel this instability in different ways. Some trajectories amplify oncogenes, others disrupt their repair pathways. And these distinct evolutionary trajectories can strongly influence therapy response. So we began to ask whether the heterogeneity in outcomes we see clinically could be explained by the heterogeneity in different evolutionary patterns. And that leads to our central question: do distinct patterns of chromosomal instability drive different evolutionary trajectories and patient outcomes in osteosarcoma? To address this, we assembled a comprehensive osteosarcoma cohort. We collected longitudinal samples from diagnosis to resection and even to metastasis from 71 patients. For both tissue biopsies and patient-derived cell lines, we performed multi-omic profiling, including whole genome sequencing, RNA-seq, ATAC-seq, Hi-C, and even spatial transcriptomics. These give us a unique dataset to connect genome instability, regulatory rewiring, and to trace the evolutionary trajectories that shape patient outcomes. So let's start with the treatment-naive stage diagnostic samples. Here, we are looking across the whole genome in 51 osteosarcoma patients. Nearly every chromosome shows alterations, with focal amplifications at MYC, CDK4, and CCNE1, and some focal deletions at tumor suppressors, including RB1 and TP53. So this illustrates the pervasive chromosomal chaos that defines this disease. But copy number variation only shows us one layer of this chaos. We then integrate across all mutation types, including single nucleotide variation, which is short as SNV, copy number variation, which is short as CNV, and structure variation, which is short for SV. So three things stand out. First, TP53 deficiency is pervasive in osteosarcoma. Second, many driver alterations are actually structure variation driven. For example, RB1 is here. Yeah. RB1, ATRX, FHIT, and LRP1B are frequently disrupted by structure variations. And third, high-level amplification of oncogenes such as MYC, CCNE1, and CDK4 is common. So this underscores the importance of whole genome sequencing, because many of these structural rearrangements-driven events will not be captured by the whole exon or panel sequencing. So taken together, osteosarcoma does have recurrent drivers, but through diverse mechanisms, making the landscape highly heterogeneous. Beyond single gene alterations, osteosarcoma is also defined by complex structural rearrangements. Across our cohort, we see chromothripsis in nearly 80% of patients, whole genome doubling in over 70%, kataegis in more than half, and ecDNAs in about 50% of the patients. Importantly, these complex structural rearrangements are detectable at diagnosis, highlighting that catastrophic genomic events occur early in osteosarcoma. To test this, we reconstruct the phylogenetic trees from paired diagnosis and metastatic samples. What we found is that the major driver alterations, including TP53 loss, RB1 disruption, MYC and cis-NE1 amplifications, are typically clonal, arising early in evolution and carried forward through clonal sweeps. In other words, by the time patients are diagnosed, the fundamental drivers are already in place. But diagnosis is only the starting point. Patients then receive chemotherapy, and we ask, how does therapy reshape the tumor genome? Across the patients, we see mutational burden increase, more loss of heterozygosity, and enrichment of SBS 35, a chemotherapy-associated mutational signature. So, chemotherapy acts as a double-edged sword, suppressing tumors while promoting further instability. Here, I'm showing you OS186, a representative case where we had samples from diagnosis, resection, and metastasis. At diagnosis, the tumor already carried MYC and cis-NE1 amplifications. After the chemotherapy, MYC remained stable, but cis-NE1 underwent massive further amplification. By the time of metastasis, cis-NE1 amplification had become even stronger, alongside new structural rearrangements. So, this case illustrates how therapy can fuel additional amplifications and reshift the clonal architecture during progression. But to better understand these structural rearrangements, we asked, how do they functionally impact the tumor? Do they just add extra copies, or do they reorganize the genome to hijack enhancers and drive oncogene expression? To move beyond this single case, we also developed a method to systematically detect structural variation-driven enhancer hijacking events across cancers. I won't go into the technical details here, but briefly, this method integrates whole genome sequencing and chromatin data to map promoter enhancer rewiring events. So using this approach in a pan-cancer dataset, we confirmed that the structural rearrangements induced enhancer hijacking is a recurrent mechanism that drives high level of oncogene expression across many cancer types. So let's back to OS186 as a case study. Here, we integrate Hi-C, ATAC-seq, and H3K27 sedation profiling in the OS186 metastatic cell line. On the left, you can see new chromatin contacts formed by the translocation event between chromosome 8 and chromosome 10, bringing strong enhancers into the proximity with the promoters of MYC, YEP1, and cis-NE1. On the right, you can see the expression data. Those genes are among the top expressed genes in the cohort, consistent with enhancer hijacking driving oncogene activation. So taken together, this illustrates how structural variations in osteosarcoma don't just create genomic chaos, they rewire gene regulation in ways that fuel tumor progression. So far, I have shown you that structural variations are pervasive in osteosarcoma, that they can expand under therapy, and that they have a real functional impact through enhanced hijacking. So given how important the structural rearrangements are, we wanted to take a step back and ask, can we summarize all these complex rearrangements into a few common evolutionary patterns? To do this, we performed a de novo extraction of structural rearrangement signatures in our diagnostic cohort. What we found is that osteosarcoma actually fall into full reproducible rearrangement signatures that are conserved from diagnosis through resection and even into metastasis. Let me start with two representative examples. On the left is an ICE3 low tumor. These tumors tend to accumulate complex translocations and chromosomal and chromothripsis-like events. On the right is an ICE3 high tumor. Here, the genome is dominated by tandem duplications that span across multiple chromosomes. These two cases illustrate very different structural variation patterns. When we scale this up across the entire cohort, these patterns don't just fall along a spectrum. Instead, they cluster into three major archetypes, as shown in the ternary plot. While there are intermediate cases in the middle, most tumors show a strong bias towards one archetype. So to put these archetypes in perspective, I like to draw a parallel with Darwin's finches. From a common ancestor, finches radiated into different ecological niches. One specialized for insects, sorry, another for, sorry, one specialized for seeds, another for insects, and another for cacti. Each archetype represents an adaptive solution, but also comes with trade-offs. So in the same way, osteosarcoma genomes radiated into three distinct evolutionary archetypes, and each archetype provides a unique route to sustain tumor growth, but at the cost of other dimensions. So, what are the trade-offs that define these three archetypes? When we annotate them by a known structural rearrangement-driven process, we see very distinct associations. Archetype 1 is strongly linked to the whole genome doubling. Archetype 2 is enriched for kataegis, a localized hypermutation phenomenon. And archetype 3 is tightly associated with chromothripsis. So each archetype is defined by a catastrophic process, and that's the trade-off, because choosing one route to genome instability will limit the options for choosing the others. Among the three archetypes, ICE3 really stood out, because ICE1 and ICE2 represent more familiar catastrophic events. But ICE3 is defined by whole genome doubling, a very different evolutionary strategy. Because whole genome doubling can both destabilize and stabilize the genome, we decided to take a closer look at ICE3, starting with its link to TP53 On the left, you can see that I3 high tumors are significantly enriched for TP53 disruption. And that makes sense because losing TP53 function lowers the barrier to genome doubling and less unstable cells to survive. And importantly, at the DNA level, mutation analysis shows that genes altered in IS3 are significantly enriched in DNA repair pathways, reinforcing the idea that these tumors have impaired repair capacity. We next ask, what does I3 actually look like at the structural variant level? On this slide, you can see that I3 high patients are specially enriched for single events, especially tandem duplications and deletions compared to I3 low. So what could be driving the massive number of tandem duplications in I3 high tumors? A simple guess is HRD, which is short for homologous recombination deficiency. When we correlated I3 high burden with the SCAR HRD score, a measure of homologous recombination deficiency, we found a strong positive correlation. We validate this association through 3 orthogonal measures of HRD. In summary, I3 high tumors exhibit increase of loss of heterozygosity detected from the HR detect. More microhomology associated indel signatures and enrichment of breast cancer HRD signature. So all of these point to HR repair deficiency. And functionally, when we tested patient-derived models, I3 high tumors showed a trend toward increased sensitivity to PARP inhibitors, though the statistical significance is limited by the sample size. So, what type of HRD might I3 represent? Is it more BRCA1 or BRCA2? Unfortunately, at the DNA level, there is no recurrent BRCA1 or BRCA2 mutations in I3 high tumors. However, at the RNA level, I3 high tumors show downregulation of homology-directed repair pathways, suggesting this might be a non-canonical HRD. We therefore performed ATAC-seq from patient-derived cell lines. And what we found is that there is a significant reduced chromatin accessibility at the BRCA1 promoter, but not at the BRCA2 promoter. So taken together, this provides direct evidence that I3 high tumors exhibit a non-canonical BRCA1 HRD-like profile. So far, I have shown you that I3 high tumors exhibit non-canonical HRD and whole genome doubling, largely driven by TP53 disruption. And as a result, these tumors are enriched for single tandem duplications and deletions. But the molecular mechanism underlying this specific pattern is still unclear. A likely mechanism is the replication stress. So in the cell cycle, when the replication fork encounters an active transcription unit, it can generate R loops and double-strand breaks. These breaks are then misrepaired, producing tandem duplications. Indeed, first, we observe a strong enrichment of single tandem duplications at the replication region. Second, when we overlay additional signals, tandem duplication regions are strongly enriched for R loops, RNA polymerase II occupancy, and active enhancers marked by H3K27 acetylation, suggesting an active transcription. Altogether, this supports a model where transcription-replication collision drives the accumulation of tandem duplications in I3 high tumors. So next, how about single deletions? Here, we see a very similar story. I3 high tumors accumulate significantly more deletions at common fragile sites, like LS AMP, DLG2, and CNTN-AP2. These regions replicate late and have fewer replication origins. So under high replication stress, they often fail to finish replication and get lost as deletions. This means I3 high tumors are shaped not only by tandem duplications, but also by deletions at fragile sites, both consequences of strong replication stress during the cell cycle. So I3 high tumors are not only defined by tandem duplications, but also by recurrent deletions at fragile sites. Those are two sides of the same coin of replication stress. Now we have a model that explains the spectrum of structural rearrangement events in I3 high tumors. But one key puzzle remains. When do these events actually occur? In normal bone development, cells smoothly progress from mesenchymal stem cells into mature osteoblasts. But in osteosarcoma, this differentiation process is disrupted. So we ask, at which stage are mutations accumulating? Here you can see that in both I3 high and low tumors, mutations are significantly enriched in osteoblast precursors. This suggests that the precursors are a weak spot during bone development. They are dividing and differentiating at the same time, which makes them especially prone to replication stress and genome instability. So now we have a full model to illustrate the evolutionary trajectories of I3 high tumors. With this model in hand, the next question is, does I3 status matter clinically? In specific, we wanted to ask, can it predict patients' prognosis? Here, you can see in the discovery cohort, patients with I3 high tumors had much worse survival outcome compared to I3 low, with a hazard ratio of 4.6. By contrast, by contrast, the HRD categories, which are prognostic in breast and ovarian cancers, were not predictive in osteosarcoma. And on the right, multivariate analysis shows that eye status remains the only significant predictor independent of age, sex, or HRD status. So this de novo rearrangement signature captures a clinically relevant axis of prognosis in osteosarcoma, while canonical HRD categories fail. We further validated the prognostic role of our I3 signature in two independent validation cohorts. One cohort is composed of MD Anderson cohort, MD Anderson full cohort, and the Sloan-Kettering cohort. And in both two validation cohorts, patients with high I3 score tend to have a significant lower survival outcome compared to the patients with low I3 score. And this association is independent of clinical covariates, for example, age and gender. So far, I have shown you that I3 signatures predict prognosis, but tumors are not just cancer cells. They exist within a complex ecosystem of immune and stromal components. These interactions may help explain why I3 high patients do so poorly in terms of a survival outcome. So our ongoing work is to dissect this tumor microenvironment using spatial transcriptomics, and I look forward to sharing those insights in the near future. So in the end, I would like to end by acknowledging the people who made this work possible. So first, my mentor, Christina Curtis, for her guidance and support. I am grateful to members of the Curtis Lab, especially Clemensons, San-Yung, and Liz for their direct contributions. Thanks also to Dr. Hobbit for her pathology and clinical expertise, and to Dr. Sweet Cordero's lab, including Leanne, Elizabeth, and Angel for their collaboration. Finally, I want to thank the Osteosarcoma Institute, PacBio, and the Stanford Cancer Institute for their support. And thank you all for your attention.
Thank you so much, Dr. Zhao, for a super thought-provoking presentation. Everyone, if you have any questions, please feel free to add those to the Q&A, and we'll share those with Dr. Zhao. And actually, maybe before you stop sharing your slides, if you wouldn't mind going back, I had a couple questions to kick it off, about, you had a slide that had it kind of showed the three archetypes. It was like the triangle, and it kind of showed them clustered.
Yes.
This one, yes. So I know you were focusing your discussion on the RS3, but I was just curious, what's the breakdown of, like, percentage-wise of RS of the three different archetypes?
Well,
in your sample of the 71.
Yeah. So I think. Sorry. So I think this will make sense. Okay. I think go back to this. So clearly, you can see around, like, I think more than one third of the patients are driving by the I3, and that's why their survival is very poor. And I think for ICE1, I think they are still like 1/3, and ICE2 combined with ICE4 are like 1/3. Yeah.
Okay. And then if you go back to that next slide, the one with the, yeah. So it looks like, just maybe if you can talk a little about like any plasticity in these signatures. I mean, it looks like, or any difference between primary and mets. It looks like in your diagram, it's kind of the same, that they don't change from one to the other.
Yes, and this is the point that we want to highlight, is because so the de novo signature fitting was based on the training of diagnostic samples, but we found that this archetype is persistent, not only in the diagnostic samples but also in resection and metastasis samples. So this means like even the microenvironment between the metastatic lesion and the primary lesion are very different, but the evolutionary path in terms of the osteosarcoma genome is very similar. So that's why the both fall into the like similar archetypes shape. Yeah.
And I thought also the slides you had about the chemotherapy mutational signatures and how that changed was really interesting. And so what about, so even therapy, like can the signatures change after like subsequent therapy after MAP?
Well, so okay, so I think here is I would say a little bit biased here because all the metastatic samples that we acquired, they are from post chemo treated samples. So that means those samples are resistant to chemotherapy in nature. Otherwise, we won't be able to get the samples. And so for those patients, of course, if those patients were treated by chemotherapy, that will increase their genome instability. But for the patients, some of the patients, their tumors might be gone after receiving a chemotherapy. So in those patients, we don't have the chance to get their tumors because their tumors are not there. Yeah.
But did any of these patients have, after they went through multiple rounds of therapy, but they're still staying in their archetype?
Well, yes. I think after patients were treated for those patients, even though they were treated by different combinations of chemotherapy, their archetype remained very similar. So it won't change their archetypes after being treated by the chemo. Yeah.
And so you're saying that you can tell the archetypes are established already at diagnosis. And so I'm kind of thinking about clinical translation. And if I guess, first of all, first question, like in terms of clinical translation, is there a feasible way to kind of determine what archetype a patient falls into at diagnosis to perhaps determine kind of what their first line therapy should be? Because I was so interested in the example that you gave about MAP and was it cisplatin that increased like the CCNE1 amplification? And so depending on kind of, what that patient looked like, maybe you should be choosing a different type of initial chemotherapy.
Yes. Yes. I think, I think so. First, yes, I think one alternative way we are pushing this, we are pushing the clinical amplification. Sorry, we are pushing the clinical application of this signature is once patient get diagnosed, we're gonna perform the whole genome sequencing by using the tissue biopsy and the peripheral blood to help us to define their I3 archetypes. So once we have their archetypes being assigned, we already performed the drug screening in the patient-derived cell lines to screening out for those drugs that might have some therapeutic event. So for patients with I3 low, I think the regular chemotherapy will work. But for patients with I3 high, it seems like the chemo won't work. So that's why we need the drug screening to identify the best drug that could be used for treat this group of patients. Second, we also understand doing the whole genome sequencing might be super expensive. So another effort in the lab is trying to pushing the cell-free DNA detection. So in other words, it's could we just use the cell-free DNA from the patient peripheral blood to determine their RS3 subtypes. And third, back to your question about this specific OS186. Actually, I think our drug screening data has shown that. So basically, first, chemotherapy is not working in this patient. But this patient, because of its super high I3 signature, so this patient is sensitive to the PARP inhibitors. So that might be an alternative treatment strategy for those chemo-resistant patients.
Yeah, that's a really good example. And does it matter? Is it in combination with, or is it after chemotherapy?
So actually, our current data only shows single dose drug screening. I think in Cordero's lab, I think Lian has another paper going to be out talking about using the combination drug screening for the osteosarcoma patients.
Love it. If anyone has any questions for Dr. Zhao, please feel free to put them in the Q&A and we can share those with him. And so I know that each of those archetypes you said were related to a specific type of catastrophic event, whether it was a whole, RS3 was a whole genome doubling, and RS2 was chromothripsis, and RS1 was a kataegis. But they weren't mutually exclusive, right? So did you see, were there any, and maybe more interestingly within that RS3, which you kind of identified as the patients that likely wouldn't respond to chemotherapy, were there any other patterns between, they were mainly whole genome doubling, but did you see any difference if they also had other events happening?
Well, so first, ideally they should be mutually exclusive. But at the same time, we also observed I3 high patients, some of them is also enriched for ecDNAs. Mm-hmm. So for example, I think this patient gonna be also OS186. So this patient is I3 high, but we also detected the ecDNA as diagnosis and metastasis. And our Hi-C plot also suggests there is a promoter-enhancing interactions between the oncogenes, across oncogenes on the ecDNA. So yes, there are some heterogeneity within the whole genome doubling group archetype, but still we are trying to try to understand it. Yeah, could we further subcluster those I3 high patients? Yeah.
Great. And then we have a question: does the RS3 signature predict sites of metastasis?
We haven't tested yet. And I think 80% of our patients have non-mets. So will be interesting to test in the future if we have more like matched primary and mets.
And so what will you be focusing on next after this? What are the next burning answer?
I think, well, you mean the question is like, what will be the next step for this project?
Yeah.
Oh, okay. So actually we just built a linkage between those structural rearrangements and the tumor evolution. But we know tumor is a microenvironment. So the next step is to estimate the consequences of having this I3 signature in terms of reshaping the tumor microenvironment, and to further investigate how will tumor microenvironment change in combined with I3 high signature will be a more robust prognostic marker for osteosarcoma patients.
Fantastic. All right. Well, if we don't have any other questions, thank you so much for spending time with us today, Dr. Zhao. Really appreciate it.
Thank you so much.
And thank you for all you do to make it better for osteosarcoma patients. More information on this and all osteobites can be found on our website at mibagents.org and on our MIB Agents YouTube channel or at your favorite podcast place. We have a new episode of our AYA podcast Osteo dropping next week on September 18th. Our co-hosts Mia and Sammy, along with other osteo warriors, will discuss turning pain into purpose, their powerful journey from patient to patient advocate, which is really apropos for our September Childhood Cancer Awareness Month. And then we'll be back with Osteobites on September 25th. We are going to be joined by Dr. Amy Armstrong from Siteman Kids. She's going to be discussing an open label cohort sequential dose escalation and dose confirmation phase 1-2 clinical trial to evaluate the safety and efficacy of dometinostat in combination with sirolimus in adolescents and adults with relapsed refractory sarcoma and osteosarcoma. You can find our Osteobites lineup for the next few months on our website. You can subscribe to our YouTube channel to never miss an episode. And if you have ideas for future topics that you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Zhao, for spending time with us today. And thanks to all of you for taking time out of your busy day to join us. We hope to see you back here on September 25th when we chat with Dr. Amy Armstrong. Thank you. Thanks, Dr. Zhao. Thanks, everyone.
Thank you so much.